Yes.
Good? Yeah.
Yeah.
Well, so I'm currently in Australia, but this work was done back in 2016 through 2020. It was a byproduct of my master's actually, and in which the idea was totally different. It wasn't proposed to be a package. It was supposed to be just exploring the method, which I'm going to talk about. And it took me a few years after the master's to get it out and make it a package and make it a paper that's usable for everyone. But that has nothing to do with what I'm doing now. Now I'm doing a PhD in noise pollution in Australia, in Brisbane, at the Queensland University of Technology. And so it's very different from the package itself and from SoundShape. And I gotta say straight away that I haven't done much since I've published it. It's out there. I'm still curious to see what's gonna do for it and how people's gonna use this. And I'm open to talk about it. So on to it. So today I'm gonna talk with you guys a little bit about the. Sorry, just a second. So I'm gonna give you guys a little introduction, talk a little bit about the Eigen sound method and the SoundShape package, and then we open up for questions. So very brief introduction about it, about bioacoustics really, and I feel like I'm talking to people who know this, but bioacoustics, it's mainly the study of animal sounds. That's how I like to think about it. And the reason we study animal sounds is because we can identify them. So if you're out in outdoors place, you can hear birds, you can hear crickets, you can hear mammals, and by listening to them, we can identify which ones are roughly the group species at least. And this is because each species has different parameters of their calls, which enhance their communication within species and between species. So these parameters are actually what we are interested as scientists. And in the last few decades, these bioacoustic parameters have been used to mostly for taxonomy at first, in which different species have different calls, which helps with the identification within each species. So we use these parameters, and this helps identify cryptic species and complex of species, and has been widely used, especially through integrative taxonomy in the past few decades, maybe. But also with the advances of the autonomous recorders, which would just gather hundreds of hours of data at a time, the bioacoustics has gained a very good impulse of this because it presents a good opportunity for conservation and just less hours of man work and more hours in the lab. And to the point where we are now looking at using bioacoustics to monitor populations and identifying individuals and estimating sizes of populations. And now with the advances in machine learning, we are just being able slowly to just identify the species without even listening to their calls. Kind of, as long as you have a data set, you can train it. We are very deep into this, so it's still a very promising field of study. But it has to be said that most of these studies, they're actually relying on the same parameters. And these parameters can be roughly divided into the frequency ones and the temporal ones. So the frequency ones, they are the one we are measuring at the spectrogram figure. We're going to talk a little bit more about this later. But the main ones would be like the peak frequencies, the dominant frequency, the carrier frequency, which are the highest points of energy. The bandwidth, which is how broad it is, how many frequencies that call encompass. And if it has a modulation, if it increases the frequency, if it decreases the frequency during the call. These are just the main ones. And the temporal ones are actually the durations of things, like could be the duration of the signal, could be the duration of the whole call, or even the numbers of calls during in each call, the intervals, and even if you get an amplitude modulation. These are roughly the ones. And these ones can actually be divided into static versus dynamic if you consider how much they vary within each species. So if you go back and look at the time parameters, especially, they are the ones often most variable. And this is because they're often related to the temperature, they're often related to how does that specific species cope with the environment. So if you get it, for instance, a frog crawling alone in a swamp, kind of, he might not be that excited. He might just give a call or two and just be quiet. But if you get several males, he might be more interested. So he might make more calls. He might make calls with shorter intervals or longer durations, or even he could change the pulse numbers. And whereas the frequency parameters are often more restrained to the calling apparatus of each species, for instance. So if I go back here, the frequency will be related to the larynx, to the vocal sac, to the vocal sac aperture. So that all sums up to just give them that vocal signature, which is what it identifies the species within each species. So the temporal parameters are often more dynamic, whereas the frequency ones, the pulse rates are often more static, which is what makes each species identify themselves within the species. And I'm talking about the frog here, but it could be the cricket, and in which you get the anatomy of the leg is what gives it the pulse rates and the shapes of the calls. So this what we have to look at is actually the statics and less of the dynamics. Whereas what we are actually currently doing, we are actually looking more at the temporal ones. So most of the parameters are temporal, which are often the most variable ones. So the studies that actually looked at more into the anatomy of how does this sound is produced actually found some strong phylogenetic relationships. So you get example of this within the frogs, in which you consider a note as calling it the cycle of inhale and exhale, considering the anatomy of it, and then you get some examples of good phylogenetic signals. You can get this from cicadas, crickets, same thing. If you look at the anatomy, you get some interesting results. Whereas for birds, it can be a bit more complicated because they have more complex calls, and it's often not as easy to identify the acoustic units, the homologous comparison. So what we often do, what has been done, is that they use the social contexts of the calls, and that's how you identify them, how you map them through an evolutionary perspective. And we find this a similar situation is the comparison between the morphometrics and the bioacoustics, in the sense that the traditional morphometrics is also reliant on just the straight up measures, which are the lengths and the numbers, and all very straight up. And if you just apply this to taxonomy, it sometimes you try to diagnose two species based on the foot length, and which can be tricky because you get lots of variation. It's just a straight up measure and doesn't really reflect how is the shape of that animal, which is the same for bioacoustics. We are just making straight up measures and counting things and giving values that doesn't necessarily represent how is that call, how is the shape of that call, what is the information he's actually carrying. So in the morphometrics, the alternative to this is to consider the geometric morphometrics approach, which is, instead of looking at straight up lines, we are actually looking at the anatomical landmarks, which are supposed to be homologous between species. And so we are looking at very stable formations and relationships between species and within species. So here in the fish example, where the fin is attached to the body, it's homologous between species. So you can actually plot this in a matrix and use this and assess this variation. How does the shape of that fish changes between and within species? And then you can ordinate and use some discriminatory analysis to see how does this variation spread across species. So it's a different way to look at the anatomy, to look at how the shape of it changes instead of using just straight lines. So back in 2013, Norman MacLeod did something very similar for bioacoustics. He employed some geometric morphometrics approaches to signal analysis from mammal biology. And what he did, he just gathered a group of bat species from the UK, in which you get two species, the Myoides ones, which are kind of famous from being indistinguishable bioacoustically speaking, which is not so good for the conservation. So what he did, he just looked at the spectrograms using a geometric morphometrics approach. And I'm going to talk a little more about this, but he managed to do it without the traditional parameters. With using the geometric morphometrics, he got some very interesting results. However, Norman did not really mention how he did it. His paper is very good, very complete, and it's a lot of information, a lot of background theories, all very well based. But he didn't mention where to do it. He just left that as a blank space. And I've talked to him about it later on. He didn't. Maybe he forgot, but he didn't use it. He didn't mention where to do it. So that's where I came in. Let's try to replicate this. Let's try to code this analysis and make it accessible for everyone. And that's what I did. So I developed the SoundShape package, which is supposed to use the methods from MacLeod, but into our platform, which is free and accessible for everyone. So that was my main goal when developing the SoundShape analysis. The SoundShape analysis, so it's based on the Eigen sound method from Norman. And in order to understand the method, we have to look back and understand how is the sound represented graphically in the way we do it more in our modern age. And again, I feel like I'm talking to people who know this already, but just in case, so the sound behaves in the nature as a wave. And what that means is if you get a speaker, which can be anything, this is how it works. It just creates compression versus vacuum waves in the air by spreading the energy across through a point across the space. And that can be translated into a sound pressure against time, which is how we are used to seeing the wavelength, the sound waves. So the frequency in which this speaker creates the waves, which is given in hertz, which is a measure of cycles per second, it's a frequency. So if you get a very simple wave in which this frequency is constant, and you translate this into a power spectrum, you get the power in one axis and the frequencies. You can see how is that energy distributed across different frequencies. So if you get a tonal wave, as a very simple one, in which that frequency is constant, you get a single value of frequency. But if you get a sinusoidal wave in which you get multiple frequencies combined, if you decompose this, you're actually going to get the energy distributed through different frequencies. But in real life, the waves are actually quite complex. So what we have to do is actually decompose it in several power frequencies that shows us how is that across sound wave. And if you do this and get enough power spectrums, and then you just rotate them, stack them off, and add an X and Z axis to the energy, you get a three-dimensional graph in which you get the X axis as a time. So you can see how is that energy varying across time. You get the Y axis for frequency. So you see how the energy is distributed across the frequencies. And the Z axis is just the energy. So if we focus on the Z axis and we add actually a scale to it and add some colors to it, and if we do this enough times, we actually get a three-dimensional graph that is very complex and represents how is that sound wave in a three-dimensional way. The way I like to look at this, it's just like looking at the picture of a mountain, in which the red here, the hot areas, it's where the most energy is gathered, and that just spreads around through the valley and gets fainter across the background noise, where the information is kind of lost. But if you have to look at this always three-dimensionally, it's not that useful. So we need a new way to do this. And it's the same as looking at the pictures of mountains. You don't see pictures of mountains like this. You see pictures of mountains from above in 2 dimensions, in which the third dimension is actually here. It's just the scale. So whenever we look at the spectrogram figure, which is now something we are more used to, we are actually looking at a 3-dimensional figure, in which the red areas are the highest peaks, and you get the valleys of background noise where the energy faints. So that's what the Eigensound method does. It compares between these spectrogram figures, just considering them as 3-dimensional figures. So if you look at this and consider it as 3-dimensional, it's too much data. If you go into it and go into spectrogram figures, it's just too much data. It's a very extensive and redundant figure, which is supposed to be redundant to create the good resolution we see in figures. But that's not applicable for the Eigensound method. We needed to reduce this amount of data. And the way that the method does is by creating a sampling grid. So you get that 3-dimensional figure and you add a grid, which is going to depend on the subject and the study and how much detail do you want. But regardless of how dense this grid is, and it's supposed to be less dense than the original matrix, it's always the same grid. You have to have the same time length, the same frequency band, and the same number of grids in each direction. And by doing this, you actually get the intersections to be always the same. So you always get the same X values, the same Y values. And what's varying is just the Z values, which are gathered at the intersections of each of these points, of each of the line grids. And by doing this, you simplify the 3-dimensional figure. And you gather some semi-landmarks, which are points that can be compared later on. And these 3-dimensional points are considered to be homologous in this context. If you use the same windows, the same X length and the same Y length, these points can be compared across. However, how about the background noise? It could influence, doesn't it? It could affect the positioning of the semi-landmarks. So the method has to account for this. So the way to think about it, at least the way I like to think about it, is, as I mentioned, is just like looking at pictures of mountains, right? In which you get the top of the mountains, but the bottom of it is as if it was covered with fog. Can you picture this? It's like only the tip of the mountain is you can show, but the valley you can't really show because it's mixed up with background noise and you can't really measure that. But the peaks of the mountains are always there, pictured, and the sound wave is always the same. So what we do is actually look at the mountains backwards. Instead of looking at how high it is, you just make a relative measure from the highest point to a point which minimizes the background noise. So in this study, I just consider it to be 25 dB, the relative measure from the peak to the lowest point, and then you create a curve of relative amplitude. And by doing this, so here you can see the background noise here, so it is affecting the image. So if you get the curve of relative amplitude, you minimize the noise from the background, but you still retain the main shape of the call, which what we are looking at. So by doing the curve of relative amplitude, we just minimize the effects of the background noise. But that's not enough, because if you look at this image, just focus on the right one here. If you place this very same one on the very right end of the window, and you compare just the two of them, identical figures, you're gonna get different results just because they're placed in different places. You won't get this in the y-axis because then you're gonna get a frequency difference. But in order to restrain the variation in the x-axis, which is in the time domain, we need to actually align these figures in the left, align them at time equals 0. And by aligning them at the left side of the window, any variation you get on the x-axis will be due to changes in the call duration. So you want this variation. And by doing like this, by aligning the units and focusing on the peaks of amplitude, you get a comparable way of looking at spectrograms, but just looking at this same landmarks. So these ones here are comparable already. And that's what Norman did. By doing exactly like this, he managed to study the meiotis bats, and he managed to get some interesting results. So in the upper graphs here, the discriminatory analysis, if you focus just on the blue and purple, you see that there is a big overlap here, which is the meiotis paths. And this is using just the traditional parameters, the ones from literature, well, the ones we're used to, the peak frequencies, that determine the durations of calls. By doing this, he could not discriminate between species. But once he applied the Eigensound method, he actually got them separated. So the Eigensound method actually managed to gather enough information to separate the species. But Norman did not account for something, which is the calls from different durations. From his sample, if you look at it, most species have a very similar duration of calls, except this one, which has a long one, but all other ones have very similar durations. But sometimes that's not true. And sometimes when you look at your data, you're going to get some very small ones and some that are very long. And sometimes you get several species with small ones and others with longer ones. So by doing this, and that's something I've learned empirically just testing the method, is that if you get several species with short calls, they will be gathered together based on the durations of their calls and it will be hard to separate between them. So what the SoundShape method adds to the Eigensound method is that we can add a log scale on the time axis. And by adding a log scale on the time axis, you gather a lot more information from the short duration calls, but you also get information from the long duration ones. You're not discarding them. You're just getting more information from the shorter ones. That has not been extensively tested, but from what I've tested, it really works very good. If you get a very various sample, kind of, if you have lots of small calls and also some long ones, you probably want the log scale. Otherwise, just the linear scale just works very good for most cases. So on top of this, the SoundShape package also helps you with the PCA analysis, in the sense that it just creates a background and there's some functions for plotting the results. And the very final bit is that it creates this hypothetical 3D surfaces that represents this variation across the principal components. And what that means is when you look at the PCA along with the hypothetical shapes, it helps you understand how is that variation across the species. So here in the example, you see that here the red dots, they have a very broad band, longer calls, no harmonic structure, which is basically the call from the centralis sample. Whereas if you get here the maximum PC1, which is the blue dots here, right across here, so these blue dots here, they would be represented by this shape, which is basically the shape that we see at Croyeri. So it really works, and it helps you understand how is that variation across species and how is your data being expressed? How is that variation across species or between species, depending on your question? So that's it. That's the SoundShape package, and that's the SoundShape workflow to it. So the paper itself and explains how to do this and gives you a workflow, a step-by-step guide with brief introductions and brief descriptions of it, so that you understand the steps to go and how to do it. And that all starts with just defining your acoustic units, because how you define them as homologous is pivotal in this type of study. And then just goes from technicalities of just creating Stopia folders and creating the wave files, avoiding the bias results, defining the sampling grid, running the PCA, and just interpreting them. So the paper is very short, but if you want more, just go into the supplementary file, because then you get the same workflow, but with lots more data, lots more information, lots more codes, and just explaining how it is done and how does it work. So that's basically it. That's not too much, but that's basically the SoundShape package, that's where I stopped, and that's where I'm curious to see where you guys go in.
Awesome. Thank you very much. Applause and questions from the audience.
Yeah, can I ask a question?
Yeah, sure.
Hi, thank you very much for this very interesting talk. Sounds very interesting. I just have a little difficulty understanding so the difference between the regular spectrogram that we use. So as I understand, what you're saying is that you have this, so the spectrogram, if you just look at it in a 3D perspective, so you will have in the z-axis, you have these amplitudes. And then you have this kind of terrain, right? And which depends on amplitudes. So in a normal spectrogram, then you have these amplitudes which you save, right, for different frequencies, and that is what you then use as features, right, if you want to use them. And here, what you are doing is that you're transforming it to a point cloud space. This is something that I had difficulties understanding. So are you so what is the data that you're storing here? So the data is it some three-dimensional points in space?
That's exactly it. It's just points in space. That's how you do it, and you get the spectrogram. It's the same spectrogram you used to use it in. I don't know if you guys use in Raven or Audacity or even in R. It's the same spectrogram. It's the same analysis in that sense. But instead of just making measures of it, just straight measures, you just looking at the three-dimensional figure and gathering points, which are homologous between spectrograms. It's a way of looking at spectrograms in a homologous ways and compare it homologously by using points. So your data is actually points. It's just point coordinates. It's much like a GIS analysis, kind of, in the sense of just looking at values.
But when we normalize a spectrogram, doesn't it also make it homologous? Because then we can also compare it, right?
That depends on what is your objective. Because normalizing the spectrogram is just changing the values of amplitude. But we are not really looking at the values of amplitude. We're looking at the relative amplitude, remember? Because the amplitude, it depends on the enthusiasm of the color. It depends on the distance. Depends on if there was like a bush between you and the animal that influences the values of the amplitude. So the values of amplitude are not reliable, but the relative values are. So within this, the recording within the spectrogram, it's reliable as long as you just consider a relative measure. So and the way that the method does is just going backwards, just going from the peak to a value below. And that will depend. If you get a very noisy recording, you might just get a very small window, a very small gap. But that variation is enough. It's just, you're looking at just at the tips of it's just like looking at the dominant frequencies, but with more emphasis to it, which are the more static parameters, right? So this is where I think the method has a lot of value, because instead of looking at the things that vary a lot, we're just looking at the things that don't vary as much, if you know what I mean.
Mm-hmm. And then you have some kind of ratios that you store, right, between different points, like some like if you have 2 points, so it kind of it finds some patterns within the spectrogram that are relative, like 2 points, like for instance the ratio between 2 points or something, right? Is that right?
It's not even a ratio really, it's just the values. Actually, we are looking at how does the values vary between the spectrograms. How is that variation across spectrograms? Maybe if I share that again.
Yeah. Yeah, I just have a hard time understanding how would that be different than what we already do with the spectro, because there also we compare different spectrograms, and there also we like try to see what is the difference in the structure, like the frequency structure, or how the amplitude varies along different frequencies.
It's just more data. If you're looking like, can you guys see this?
Yeah, we can see that very clearly.
So in a traditional way of analyzing this, you would be getting these measures just straight here, just straight here. Here, each point of this, it's a variable. So instead of working with like 10 measurements, you're just working with hundreds. So it's more detailed to it. You're not making straight measures. If you were to do a traditional analysis and you had to have a dominant frequency, it would be just one value, right? You can get a range, you can get a statistics, you can get a deviation to it, but it's just one value. That's what you're looking at. Here, you're just getting that frequency, that dominant frequency, and just getting hundreds of measures to it. So you get and by looking at these points, the sampling grid, it's homologous between them. So if you get any variation in the positioning of these points, they're going to show, and they show. And when you do a PCR with them, it shows, it's gathering the variation.
So I understand you're saying that if you calculate these features on top of spectrograms, like dominant frequencies or time interval between different points, yes, those are single points, and but when we have a full spectrogram, which is basically we have a whole time series of it, then also we get multiple, we get a lot of data points, right, long time, and then when we feed that into a model, isn't that also similar to this?
Yes and no. It all depends on what you aiming to achieve with this, and that's where I think the method. Because to be very honest, I don't think that by doing this you're gonna get very much different from doing a taxonomy approach to it. I don't feel like this is such a big gain in related, because the taxonomy is well established, isn't it? It's the way we do it. It's the way we do it. And what's the point of reinventing the wheel, kind of? But you mentioned for doing models, and I know I don't have a lot of experience, but from what I've read and from what I've talked, I know that we can gain from it. I think by emphasizing the units, and I feel like this is something that's going to be very good for training the algorithms.
Yeah.
Instead of just getting because for training you just get this couple of parameters, the 10 parameters, whatever that you use for training. Instead of getting those 10 parameters, just get a sample of 10 nodes or 10 acoustic units from that species, get the sound shape for it, get the data, get the same landmarks, and tell the model, this is my species, and just run. This is how I think it's gonna add a lot to it. Not really for taxonomy, to be honest.
All right. Thank you.
You're welcome.
Watching this, I was wondering computationally how it's similar or different to dynamic time warping. Is that
Oh, I don't know what is that you said.
Yeah, and I don't know in computational detail, dynamic time warping is where you take a sound, you're comparing sounds, but you add a certain amount of stretch. So I guess it's just in the time axis as opposed to the frequency axis. But I don't know what the computational process is there. I was wondering whether it's similar or whether this is a different.
Well, I'm not familiar with this method you're mentioning, so I wouldn't be able to tell, to be honest.
Sounds good.
What I can tell is that here you are looking at the matrix of spectrogram values. It's a matrix of amplitude values, basically. That's as far as I can. From the computational perspective, that's what you're looking at. You just computate the spectrogram and simplify it and just focus on less values so that the analysis go faster. You could just get as long as you do with the same spectrogram parameters, you could even use just a straight up matrix of spectrogram values, but that would be massive and it's going to take a lot of time.
Thanks for the presentation and the really clear walkthrough that you gave. I was wondering, in your introduction, you talked about phylogenetic studies that have been done for amphibians and insects and birds, but for birds, it was, I guess, more complicated. I was wondering what's happened since McLeod did this, apply this method with bats, and if this SoundShape approach has been used to refine or add some more knowledge to these phylogenetic studies. Like, how has this method been put into practice? I guess it was around 10 years ago that that first paper came out. So what's been the progress or the new knowledge that's been added as a result of this approach?
Well, Ben, I've honestly, when I read the paper from McLeod back then, I thought it was going to be straight up being used by everyone, but it hasn't. And I think you guys just watched the latest on this method. I don't think there's anything else. I haven't even used it that much later since I've done it, but just mainly because just getting into a new PhD and doing other things and just couldn't get my head around it again. But not much has been done with. But for this evolutionary perspective, I know that the geometric morphometrics has a lot of ways of looking at this data. I haven't explored it, but I know there's a lot of ways to do it. Once you gather the data with the semi-landmarks from the spectrograms and do it homologously, you can do some very interesting analysis. You can do some phylogenetic analysis using geometric morphometrics now. So this is the main. So to sum up, from Norman's original work to now, what we have now is just a bridge. Is you can go from bioacoustics to geometric morphometrics. That's what SoundShape does. It just crosses this bridge, so you can go from there. But no one has done it. I'm curious to see, to be honest.
And then what about, like, what you just said made me think, what if we're not interested in a single call or a series of calls from a single species, but what if you took like 24-hour spectrograms and you wanted to do a morphometric analysis of that? And so instead of comparing species calls, you're comparing like the daily soundscapes from different places. That could also be a way of, I don't know, comparing different places, different times, habitats.
Yes, I agree. That's something someone already told me. Like, you guys probably familiar with the long duration spectrograms, a way of computing the acoustic indices and just plotting them together, and then run a SoundShape on top of that. That would be fun. I think that would be interesting. No, I think no one has done it. That's a good topic, probably a PhD.
I've seen someone do
My internet.
PSNE analysis from the images. It'd be really interesting to put those two side by side and see how similar or different the insights are, SoundShape approach and the TSME from the image data? Are those recovering similar clusters, or is it looking different features?
Yeah, I'm interested. I think it's going to be very interesting. I'm curious to see how is this going to apply. I don't know yet, but I'm curious to see.
So Pedro, thanks for the nice talk. And this is a very interesting and creative idea to think about sounds in a geometrical way, geometrical morphometric way. So when we think, we remember the ecological and evolutionary applications of geometric morphometrics. So we can imagine that within a species, different guys everywhere, all around the world, can grab individuals from the same species, and they can have the same anatomical marks that won't change, and then the variation within species then can be consistently attributed to like an ecological process or something that is determining the variation within individuals, right? So when we think about sound, we know that there's a large variation in ways we can record sounds. So the output, the analytical unity, depends on how we record that microphone, the sample rate, bit depth. So I was wondering how that would influence the within species variation in this analytical approach, because I think that, for instance, you use some few points, but is it going to? What if this is like the most purposive source of variation within species? You know.
Well, Larissa, I agree. That's where you get the most variation within species, isn't it? From how to record them. And that's why that's a tricky one. That's why the method just do this curves of relative amplitude. That's how you do this. Just that's how you compare between species and within species. Since there is so much variation in how you gather that data, this method of just focusing on a relative amplitude, because the relative amplitude doesn't depend on the recorder, doesn't depend on the distance, doesn't, as long as you get enough data on your recorder, doesn't matter which one is, as long as you get enough, so that you enough in the sense of it just goes above the background noise. So you, as long as you have minimal signal to noise ratio, I think that's a technical term, you can do it. You just look backwards. By looking backwards like this, from top to bottom, you can compare. And I think that's where you can get some very subtle results. If you get it, especially if you get good recordings, but you can compare across different recorders, across different species, different timings, different temperatures even, because you're focusing on the things that don't vary as much. If you're focusing on time domains, it would be tricky because it varies a lot. It varies on temperature, on the excitement, and it wouldn't be comparable. But by doing like this, it is comparable. You can compare. It doesn't matter which recorder you did. You could even do it with your cell phone probably if you get enough of it. And
Yeah,
you can compare. And I think for the little I've explored, the within species is very interesting. You get vocal signatures from individuals. Actually, if you get a population, you actually get each individual has his vocal signature, and the ordination plots are just shows this. It's quite refined analysis, let's say, like this kind of.
Yeah, absolutely. I have to be fair. I was thinking about when you put this these very beautiful spectrograms of Cuvieri. I was just seeing some spectrograms from passive acoustic monitoring from Fisalemus Cuvieri, and it's like, so you can imagine just that part that is more with a higher concentration of energy. Only that small part appear in the recording. Nothing else. Nothing about the harmonics of this frequency modulation. It's just the part that you have the largest concentration of energy. And that's where I still see that they are really rough, but they still somehow fixed parameters like dominant frequency, peak frequency. They still are repeatable unities that can be detected in these different sounds. So for instance, if I create a sound shape of this species on these recordings that have a great signal to noise ratio where you don't know where the species are vocalizing, they're probably very far from the recorder, I'll have a very different shape. And probably that would imply a large variation between individuals. Even though that some parts of this map would have some redundancy, like the peak frequency part, right? So then something interesting that might be useful for this approach is to check on the metrics about niche complementarity in multidimensional volume, or even 2D or 3D, where they can have like how dissimilar, how they overlap, how redundant there are parts on this volume. And then this can be another way to understand the differences between and within individuals.
Yeah, definitely. I think that's a good study. I think that's what the method is for. I think that's one of the possible studies that you can use, and I'd be keen to see what happens, what results would come out of it.
There are a couple of different points you mentioned that you think of frequency as being sort of the interesting parameter and timing as being really variable. I guess this is a comment as much as a question. The community of, I've worked on a couple different communities of insects, and I've typically thought of timing as being the really key parameter for those. It does change with temperature, but frequency does also. And there's a lot of overlap between the frequency, but if I'm looking at a spectrogram and trying to assign it to species, it's often like the interpulse interval of the insects that I'm using. So just as you're thinking about method development, it might be interesting to think of a version where time has priority or frequency has priority.
Oh yeah, that's interesting actually. That goes back in how do you define these units? Because for insects it's just different, isn't it? You've just mentioned it. So the static and versus dynamic parameters is something that we should, as bioacousticians, we should always bear in mind, because it changes. Each species is going to be different, and for insects, like you said, that sometimes the pulse intervals, the pulse rates, or something I've read already, is very static between species or within species. So that's where you should be focusing on. And that would depend on how you define your acoustic units, which is for if it is for Eigensound method or if it is for traditional analysis. How you define, I think we should always bear in mind that we should account for just retaining that variation, and just even if it is for separating species, be careful with separating species with dynamics parameters. Doesn't matter if it is time, if it is, it would depend on each species. I had a few examples and would depend on the species.
Okay, so I have a question here. I'm sorry if I'm not understanding it. Can I ask you to like explain this this slide that you have opened, like this idea of relative amplitude? Like if you have to just explain it using these spectrograms here, so how would you define relative amplitude?
Well, the relative amplitude in this case is just it's relative to what? I think maybe that's what it's getting. It's relative to the peak of amplitude. So you get the peak of amplitude if you're actually focusing on the scale here. So the peak would be here, would be 0. The way we do it, since the dB levels are not reliable, you can't count on them. The values that you get it, if you go there if you go to the wave file, you're gonna see that it's just time against amplitude, against energy, how much energy through time.
Yeah.
But that value is not reliable because it depends on the recorder, depends on the enthusiasm, the distance, depends on a lot of things. So that value is not reliable. But the relative value within that, the same recording is always reliable because you get the maximum value and then you get everything backwards. So
Yeah, but
that's
this is
how we do spectrogram.
Yeah, but that's exactly what I was saying. This is something that we also do in a normal spectrogram. Like we normalize for loudness.
Yeah, right.
Exactly.
So even if you normalize, because normalizing is just getting those values that are not reliable, and you often just say that the highest one is 1. You just get it a value. This is normalizing. The highest one is 1. So then you get all the recordings and you just say that the highest one is 1.
Mhm.
And then the relative will be the same because it's going backwards.
Yeah.
So the relative will always be the same. Normalizing is like this.
Yeah. And what is the curve of relative amplitude then?
So the curve of relative amplitude. So now you normalize, let's use this word. And you've defined it experimentally, empirically, you just went through and tested.
Mhm.
And you've noticed that in your sample, all the calls that has above 25, which in this sample, but it could be 10, it could be 5, it could be 50, depends on the recording.
Sure.
From that value, above that value, it's all good. You don't get noise. The signal to noise ratio here is very good. And
Mhm.
everything below this would be, and then if you get values below this 25, you'd be
Mhm.
getting background noise.
Sure.
So the curve of relative amplitude is basically just setting up that value. It's a cutoff value. And you're just discarding all the data from below that, which just gonna gather a lot of background noise and other species maybe. So
Mhm.
you discard those values and you're just focusing on the tips.
Yeah.
So
But
if you get
the tips are still the amplitude values, right?
yeah, there's still amplitude values.
Okay.
It's this like, so
Yeah.
you had this with full spectrogram.
Yeah.
And then you set up a value and then say, I don't want these values below this. So you just discard
Yeah.
them and you
Yeah.
just stick with the tips of it. That's it.
Okay.
It's the same value. It's the same relative amplitude. It's the same relation. You're just
Mhm.
discarding the background noise.
Yeah, that's interesting. Actually, there is like for normal spectrograms that we used in audio signal processing, there is also a process called segmentation. So where we basically do something like similar, but we don't do it in this 3D way. So what we go column wise or row wise, and then we select the highest value, and then we check all the values that are below a certain threshold of this value, right? So for instance, all the values that are 2 times smaller than this. So we basically, what we are saying is that if the at a certain point the amplitude is not loud enough, then it might be noise. So it might not be the, and then we come up with a spectrogram, and that spectrogram only has values that are above a certain loudness level. And then we think that, okay, this is our signal. And then we also do some morphological operations on that image, like and join, and then in the end we get like we assume that what we are left with are like our sounds that is our signal and not the noise. So we remove the noise. And I think this might be a very like an interesting way of doing the same, like segmenting. So basically separating the noise from sound, and then only using the sound values and assuming what we get in the end are the sound values. And then, of course, this also reduces the number of data points we have. And then we can use just these data points to calculate our features, right? Because even these values that you have here can be large, like extremely large, right? I mean, in the end, the problem that we have, why we calculate features is that we want to represent sound in a way that perceptually similar sounds would generally be near in the vector space, And for that, what we do is that we don't input the whole raw data, because that would be too big, like dimensionally speaking, it would be really large and it would be difficult for a model to separate those files. So what we do is that we do some dimensionality reduction or transform it into a different, feature space. So so essentially we can do the same. Maybe it would be interesting to see. I find it interesting, so I would definitely want to use it as this segmentation process. So I initially, when I heard you speaking, I had a kind of an intuition that this is what it must be doing, but I wasn't sure about it. But I think now I'm sure that what it is doing. So thank you.
You're welcome. Good that I clarified this. It's a key point. And from what you described, it's basically the same. Is that threshold? It's basically the same.
Oh, yeah. They also use this I'm pretty sure that even in the BirdNet algorithm, they are doing this segmentation phase. There's a segmentation phase where you basically you denoise the signal in a way, and so it was there was this method, and I'm pretty sure that they're also doing it in BirdNet, the segmentation phase. But it would be interesting to see if this even actually fares better than the other method. Yeah.
Cool. Interesting.
I recognize that we've crept up on and past the hour. Are there any final questions for Pedro? Uh-oh. And Isaac is about to hit bedtime. Thank you so much for coming out this evening, this morning, this afternoon, middle of the night. This is a really fun and interesting discussion to be part of.
Well, thank you for the invitation. It's my pleasure to be here, and I'm open to it. If you guys want to send me an email, get in touch, further questions, I'm open to it.
So in case anybody wants to follow up with you.
Yeah, thank you so much for being patient with my questions.
Oh, don't worry.
I found it.
It's value. It's pretty good. I appreciate it. It's good questioning. It's what I'm here for.
Thank you. Yeah, we really appreciate it.
I hope you all have a great evening.
Yeah.
Thank you. Have a good evening, everyone.
Yeah.
And see you guys next time.
Yeah. Thank you. Take care. Bye.